Hello, everybody. Good afternoon for Portugal and good morning for our friends and colleagues in the US. My name is José Marques and I am Program Director for Medical Physics together with Dr. João Oliveira, who is the President of the Portuguese Oncology Institute. João can unfortunately not be with us this afternoon. He sends his apologies and also his best regards to everybody. As you probably know, medical physics is the most recent addition to the UT Austin, Portugal collaboration that has been quite successful for a number of years. We just started in the most recent renewal of the program, and proton therapy in particular is a topic that is of interest for Portugal, as we are planning to have a facility for proton therapy very soon. So we will have in this first set of master classes, first set for this topic, we will have 4 presentations. The first one will be done by Professor João Seco from the University of Heidelberg. João graduated with a PhD from the University of London at the Institute of Cancer Research and Royal Marsden Hospital in London. He then went to the US, to Harvard Medical School in Boston, working MMGH, Massachusetts General Hospital, and recently returned to Europe to become a full professor at the University of Heidelberg and to work at the German Cancer Research Institute, DKFZ, in Heidelberg. He is heading a new group dedicated to ion beam research, with focus on two very hot topics like novel imaging technologies to reduce Bragg peak positioning errors in patients, and research on the mechanism of radiation-triggered DNA damage via reactive oxygen species. João is also presently the chair of medical physics at the Department of Physics and Astronomy at the University of Heidelberg, and is member of the FOMP scientific committee representing the German Society for Medical Physics. João, the floor is yours.
Thank you very much, José. I would like to express my gratitude for the invitation for you and João Oliveira. I will share my screen. Is that okay? Let me see. Can you see my presentation? Can you see my presentation? I don't hear anything at the moment.
Yes.
Okay. So in that case, I would like to thank the organizing committee for the invitation, and I will be talking about the physics challenge of proton therapy. I discussed with Radhe the possibility of having 2 complementary talks. There might be some overlap between our 2 talks, but I suspect that both of us are very open to questions. So please really do ask questions when you get a chance, okay? So here goes an overview of my talk. I will give a brief introduction into proton therapy with a small historical overview. I will then discuss the advantages of protons, and then I will discuss some passive and pencil beam scanning methods. Then I will get into one of the hot topics, which is flash radiation. I'll provide a little bit of details and why. And then the next hot topic will be microbeams and mini beam irradiation. And so in both cases, I will be open to any type of questions. And if you have any comments also, it's always grateful. Okay, So the visionary person that was behind this was Robert Wilson, who proposed the idea many, many years ago in a famous paper in 1946, sorry. And in this paper, he already made comments towards many things that we use today. He mentioned that protons can be used clinically. He also mentioned that accelerators were available. He mentioned that the Bragg peak could be used to be placed inside the tumor to treat the cancer. So this is a really good idea. And he mentioned that the big advantage of protons was to spare organs to radiation. And finally, you also mentioned something called the modulator wheels, which allow you to spread out or to create energy modulation, where you can create multiple Bragg peaks to produce a spread out Bragg peak. So all this was in this initial paper, and so a lot of new ideas. So the paper was published in 1946, and within a few years, the first patient was treated in Berkeley, in California. So that's 8 years. It's a very fast development from an idea to a treatment. Then there was the first neurosurgical treatment that was done in 1958. And a follow-up, the first large field treatments were done in 1967 in Sweden. So that was a bit of a longer wait, but still, we went from small fields to large fields. So you could start to treat really large tumors. And then in Boston, at Harvard Cyclotron, they treated the first fractionated treatment. So a large field fractionated treatment at the Harvard Cyclotron. So the Harvard Cyclotron is not a hospital facility, or was not a facility. And this was done with patients from MGH. And in the first hospital facility was built in 1990 at Loma Linda. So many years from the research facility to the hospital facility. In Europe, the Proton facility, PSI, started working in 1996. Darmstadt started treating with carbon ions in 97. And finally, here in Heidelberg, the Heidelberg Ion Therapy Facility opened in 2009 and took over all the treatment of carbon and protons in Germany. So it was a big jump from protons to eventually carbon. And now in Heidelberg, we do both. There are now a lot of dual centers and a lot of proton centers worldwide. So since this is a masterclass, I want to give a slight overview of the main interactions that protons suffer when they go through matter. The most important is the electronic interaction. Basically, that means that the protons will interact with the electrons in the shell of the atom. And so you can see here an example of a drawing on the right, where the protons can either eject an electron from the atom, and this would be ionization, they ionize an electron, or they can just excite an electron inside the atom from one shell to a higher shell. So the electron does not get excited, but you have an excited atom that can break up, such as in a molecule that can have a molecule breakup. So electronic interaction is how protons lose energy. So this is the main process by which protons lose energy. Then you can have something called a nuclear interaction, and this is due to the Coulomb scattering. So a Coulomb scattering, the proton scatters off the nucleus of the atom. And this is an elastic scattering effectively and produces a small deflection. So there is no loss of energy, but there is a small deflection of theta that you can see. And usually in the case of protons, they go through many, many, many small deflections, and we usually coin that as multiple Coulomb scattering. So the concept multiple Coulomb scattering is not a physical interaction. It just represents many, many small Coulomb interactions of the protons as they go through matter. Then you have an elastic nuclear collision, which can produce a large deflection. And as the word says, elastic, there is no energy loss, but the nucleus can suffer a recoil from that. And then you can have the final, which is the non-elastic nuclear interaction. And here the main byproducts, all the fragments that you produce when the nucleus, when you break up the nucleus. Okay, so what are the advantages of protons relative to photons? I will start with the famous depth dose curves that are very well known in all the books. So on the right, you have in yellow, the photon depth dose curve for 15 MeV photon beam. And then you have the blue dotted line, which is your single Bragg peak. And the blue solid line is what is called the spread out Bragg peak. And it's made up of many single Bragg peaks. So the first thing you look at is the blue dotted line, and you see that there is an entrance, low entrance dose, which is called the plateau region. And this is usually very, very low. So this is a good advantage. You have a low entrance dose. You also have a maximum at the Bragg peak. So This is where you want to really deposit the dose. You want to put that Bragg peak in or on the edge of the tumor to take advantage of curing the of dealing with the cancer. And then you also have a very fast fall off beyond the Bragg peak, and you see that the dose goes down to 0. So this is really one of the big advantages, that rapid fall off and dose goes to 0 after the Bragg peak. And then if you want to treat large fields, you have to modulate. So you have to energy modulate. You have to put a few Bragg peaks together and you produce the dark blue line. And that is usually what is used to treat cancer. The tumors are usually larger than a few centimeters. And in this case, you need to use multiple Bragg peaks to produce the spatial spread. And finally, a good advantage of protons is they are very, very similar to photons. So there is not a huge shift in the philosophy of treatment. So in the radiation oncology, that's proton therapy. It's a relatively easy step to go into protons, so from photons to protons. And the RBE that's usually commonly used in the clinic is 1.1. Okay, so how do we usually quantify the advantage of protons? Here is a very well sigmoidal curve that represents tumor control on the y-axis versus radiation dose on the x-axis. And the sigmoidal curve basically indicates that as long as you keep increasing the dose, eventually you will achieve 100% of tumor control. That means you can give hundreds or even thousands of gray and no cancer will survive. The big problem is that in addition to giving dose to the tumor, you are giving dose to healthy organs. And so here's a sigmoidal curve in white for representing healthy organs. So as I increase the dose, I also increase the toxicity of the healthy organs. And this is really a disadvantage. Usually the white curve, the organs is usually shifted to the right because DNA repair processes in the healthy organs is better. And this gives us a small advantage that allows us also to fractionate. So when we treat, we would really like to achieve what is called a benefit point, a maximum benefit point, and that is somewhere in the middle between the red curve and the white curve. And I've represented that as the green curve. So the maximum point sits right at the tip. Right at this tip here. And this is where we would love to be all the time, try to maximize our tumor control, but also minimize the toxicity. In reality, the doctors are very scared of toxicity, and they usually don't go for this optimal point. They usually tend to go for a much lower level of toxicity in the order of 20 to 30%. So at 20 to 30% toxicity levels, I achieve around 30 to 40% of tumor control. It's not very good for some cancers, so very poor tumor control. And this is done for photon beams, If I have ability to spare organs radiation, like with protons, literally what I'm doing is moving the sigmoidal curve to the right. And so I have a significant drop in the radiation given to healthy organs, and this means that the toxicity goes down. And if I use this same curve for the same level of toxicity, I get a significant increase in tumor control. So now I'm in the 90% level of control. This is the true advantage of protons. By reducing the dose to healthy organs, I can ultimately increase the tumor control. So this is something I would like to mention, which is always present when we do proton therapy. It's a famous comment from Herman Soot, who was the director for MGH for over 30 years, who retired in 2001 or 2002, and used to say that there is no advantage whatsoever in irradiating uninvolved tissue. This is really the advantage of protons. We are reducing the irradiation of uninvolved healthy tissue. This is the key. So how do we deliver when radiation in protons? There are two main delivery methods, passive scattering and pencil beam scanning. I will describe both of them in the next few minutes. So in the case of treatment, I want to generate a Bragg peak, which is the orange curve represented here. And this Bragg peak is composed of many single Bragg peaks put together to produce this SOBP. Sorry, the SOBP is the orange curve. The green ones are the Bragg peaks. And so what you need to do is you need to be able to shift each Bragg peak in energy to the left to allow it to put these energies together to produce the spread out Bragg peak. And this is where you will want your tumor to be. And this is called energy modulation, okay? And if you use a passive scattering approach, you can either use what is called a range modulator wheel, which is this one on the right, or you could use a ridge filter. So the range modulator wheel was the system available in Boston, where I was at the Proton Center. And the rich filter is what we have here in Heidelberg at the synchrotron facility. And effectively, each little step here corresponds to a small energy absorption. And basically, it slows down the beam. And roughly the difference between each two steps is 3 millimeters. So we have a 3 millimeter step each way, 3 millimeters each time. So it's a very high resolution range control or energy control. And the whole system, the whole gantry in Boston looks something like this. You have your beam coming in, which is a very narrow beam. It suffers energy modulation here, so you have a range shift, and then the beam has to be blown up to allow for treatment. And this is done through a double scattering system, where the first scattering blows up the beam, and the second one increases the blow up and also changes the energy a little bit. And then this double scattering system produces what is called a broad beam. Here at this surface, you have a broad beam. And now the broad beam has to be shaped to the tumor. Okay. And the shaping is done with an aperture here and a compensator here. So you have an aperture which is usually made of tin, and you have a compensator which is made of plastic, PMMA or anything similar. And the shaping of a passive scattering system can only be done to shape the tumor in the distal depth. So we can only shape really well this part here, which is the deepest, but we lose the ability to shape the shallow part. And so you usually see in passive scattering systems, you have a horns, the double horns, which are very hot. But we have excellent control on the depth at the distal part. So just as an example, here is the compensator. Here's the geometry that I wish to shape with the passive scattering system. And I see excellent control over here in depth. But I have these horns which are very hot, okay? And the passive scattering system also has the disadvantage that I have to manufacture both the compensator and the aperture, the collimator, which is sitting up here. Both of them have to be manufactured. So it's an additional problem that every time I have a new patient, I have to manufacture all these components. In the case of pencil beam scattering, all these components are no longer present in the beam, and I just change the energy and I magnetically scan the shape. With the pencil beam scanning, I have the ability to really deposit those anywhere I want in the tumor, and so I have much better control in both the distal part and the proximal part, so the deeper part and the shallower part. I have no longer these horns here. So if you look carefully from the left to the right, you see this very hot area here, which is no longer here. So you lose, the horns have gone, and also this U-shaped geometry, it's relatively cooler than this one here. And the big advantage is also you have no need to build anything. You don't have to buy any compensator or any collimators. Everything is done magnetically, and the energy modulation is usually done just immediately outside the cyclotron. So the beam comes through the cyclotron onto the beam line, and you do the energy modulation directly on the beam line, so before it enters the room. Okay, so one of the hot topics at the moment is flash, and I will spend a little bit of time on this. And there will be another talk on flash, which will provide a lot more clinical information, but this one is more related to the physics. Okay, there's a fantastic review by Marco Durante in 2018 that discussed dose rate effects. And one of the most important things is to remember where it all starts. So on the x-axis, you have your dose rate in units of gray per minute. And on your y-axis, you have something called dose rate effectiveness factor. And that means a value that is very high means that there is no damage to the tissue. So a 2 represents an excellent response by the tissue to low dose radiation. And environmental doses can be anything from cosmic radiation to granite radiation coming from the stones, or any radiation coming from the walls, or anything else. Usually this environmental exposure is at 0.1 milligray per minute. So it's very, very low dose. We're at very, very low dose here. When we go into radiotherapy, the dose has to be in the order of 3 to 6 gray per minute. So this number here, 6 gray per minute, is usually the therapy system that rate. So high dose rate, stereotactic body radiation, stereostatic radiosurgery, IMRT, 3D conformal, they are all treated at 6 gray per minute. This is the dose at which there is maximum effect on the cancer. And this is the key that we use, this very key thing. So all linear accelerators, and They tend to work either at 3 gray per minute or at 6 gray per minute. Then you have the case of intraoperative radiation, which is electron beams, and they can work at a little higher, 20, 30 gray per minute, or even sometimes close to 100 gray per minute. Now, where is flash? So flash sits at roughly 2,500 gray per minute. So it's a factor of almost 1,000. So the dose rate has gone so high that now we are in the order of 2,500 gray per minute or 60 gray per second. So we are a lot more. And what's happening at flash? Well, no one really knows. And this is why I'm going to discuss this, and then I'll present some ideas. Okay, so one of the thinkings about flash is that it's related to oxygen. So here is the oxygen curve that represents the capacity of the oxygen to sensitize cells to radiation. And on the horizontal axis, you have oxygen pressure in millimeters of mercury. And on the y-axis, you have the relative sensitivity. So 1 is represented as the reference, and it represents hypoxic tumors. And as I increase the oxygen level, I start to increase the amount of cells I kill. And at around 40 millimeters of mercury, I kill 3 times more cells than hypoxic tumors. So this is a huge increase. It's a factor of 3 just with oxygen. And that increase is very quick. So just to get a factor of 2 increase, you just need to increase the oxygen by a small amount of 0.4, 0.5% of oxygen. So it is very important that tumors are oxygenated to allow us to kill the cells very quickly. So this is very important. This is called the oxygen enhancement ratio. It goes from 1 to 3. That's why when we try to treat, just 1 minute. So when we treat with radiation, it's very important for the doctor to know if the tumor is oxygenated or not. This is critical. So studies in the 60s and the 70s, they studied hamster cells and mammalian cells, HeLa cells, and the radiation. And here's a radiation survival curve of a fully oxygenated cell, hamster cells, CHO. So what is that? So on the x-axis, you have dose in rads. So 500 rads is 5 gray. So it's a factor of 100. The conversion is 1 to 100. And the survival fraction represents the amount of cells that survive radiation. So at 0 radiation, I have 100% survival. At 5 gray, for example, I have 10% survival. So this is a very quick drop at 5 gray. And this is your standard survival curve that you get with the standard hamster cells. And the same thing for the HeLa cells. At 0 gray, I have 100% survival of the cells. And as I increase the radiation, I have a very sharp fall of the radiation as a function of oxygen. Now, the funny thing they notice is that at some point, there's an inflection point if I send the radiation at flash level. So remember, I'm giving, instead of 6 gray per minute, I'm giving 60 gray per second, okay? So it's a huge increase of dose rate. And suddenly there is an inflection point where the cells have a completely different survival. So they have the same survival below this value of 5 gray as the normal oxygenated, but then at above 5 gray, they have a completely different survival. In fact, they're increasing their survival. And the same thing was observed for different types of cells. So this is a famous study from 1967 from Barry, and a previous study from Towns in 1967, this was 69, 67. They compared these curves with the fully hypoxic system. So here is a fully hypoxic cell survival curve. So it's represented with nitrogen. So the cells have no oxygen, they only have nitrogen. And here's a fully oxygenated curve, which is this line. And here is the inflection point for flash. And then flash behaves if it's fully hypoxic. So this is extraordinary. Flash is somehow removing all the oxygen in such a rate that the cell immediately behaves as hypoxic. And this is what is being called as the oxygen depletion hypothesis. And that's why it's extraordinary. So there was a lot of studies in the 60s and the 70s. He has a list of them. And unfortunately, in the case of the studies, they studied at very, very high dose rates, at 10 to 6 gray per second. This is not the case of what we're focusing on recently, but I just wanted to bring to this point that the other key information was that a minimum dose is needed to produce this flash effect. You need to give at least 5 gray at this very high dose rate. So 5 to 12 gray has to be given in this instant shot. And the rates are extremely high because these were cancer cells. So what was the revolution? The revolution started in 2014. That's almost 30 years after the studies in the 70s, 30 to 40 years afterwards. And in 2014, the group in Paris, by Fauvedon and al., they did an amazing study where they compared MEV electrons, 4 MEV electrons to X-rays in mice. And they rated lung. And what they found that is if you radiate both electrons and X-ray at the conventional dose rate, they have the same response. But if you apply flash rates to the electrons, that the toxicity in the lung drops significantly. So this is the flash rate from the electrons. And this is the conventional photon beam at 1 gray per minute or 2 gray per minute. And there is a significant increase in lung fibrosis. So here, these mice have a lot less side effects at this very high rate. So they gave the same dose with a significant drop in side effects. And they did histology also and compared, and you can see clearly here, here's the conventional, and you see this destruction of the lung tissue. While here, the control and the flash look very similar. There's some increase in toxicity, as you also see here, but still the behavior is much better than this one. This one is completely destroyed by the radiation. And that's giving 17 gray in 1 shot. So it's a very high dose. Remember that the minimum had to be above 5 to 12 gray. So 17 gray is definitely above that. So this is what launched the whole field in radiation therapy. Flash protects normal tissue. And after that, there was a significant amount of publications that have shown exactly that. These are a list of publications in a review paper in clinical oncology by Marie-Catherine Vosnin and the other Jolyn Hendry and Charles Limali. And they saw exactly the same thing. They saw a lot of sparing in intestine, foot, skin, and in mouse, in pig, in cat. And this was amazing. And usually the dose rates were in this 15, 20 gray up to 200 gray per second. So it was not the 10 to 6 gray per second, except this one. They were all moderated 10, 15, 17, 20 gray per second. Okay, so this was the clinical evidence that's starting to appear. So the funny thing is, in the case of protons, the first center to test flash in their machine was Groningen. So it was done 2 years ago, in 2019. So they took an IBA proton set machine and they flashed with it to see if it was even feasible. Although they say that the first delivery was in a clinical treatment room, they did not use flash for patients. They just used a dosimeter to check that it was possible. So this advertising is a bit misleading, but they did show that an IBA machine can treat with flash. They did not treat a patient, but they did technically show it's technologically possible. In addition, Varian have also created a very huge consortium of proton centers to do exactly the same thing, to focus on treating with flash and to develop, let's say, protocols for people to use, for doctors to use in the treatment of cancer. So to finish this section, I wanted to give a quick overview of the physics challenge. So one of the biggest physics challenges, no one really knows why flash protects organs. And this is one of the key challenges that I think will be addressed in the next few years. And the other key challenge is, how do we use the flash in the clinic? All the clinical studies have been based on single beam treatments, either a single beam electron or a single beam protons. How do we use clinically multiple beams? No one has an idea how to do that. So these are the true challenges that will be faced in the next 5, 6 years, probably longer. Okay, moving into mini beams and micro beams. So this is another way of delivering radiation that also protects organs. So the main idea is that when you treat with a standard radiation therapy, you deliver a homogeneous distribution of dose, which is this flat step here. In the case of mini beam microbeams, the philosophy is not to do that, but to deliver a spatially fractionated beam. So you have this up and down, you have this peak and valley dose. So the peaks are the top part and the valley is the bottom part. And this peak to valley dose creates a zigzag, right? Like a checker box, a black and white and stuff like this. And it's amazing, but this really works. No one really, well, we have a potential mechanism now, but it's amazing that this really works. And what happens is, usually you develop, you give a very high dose to the peak and you give a very low dose to the valley. And a simple rule of thumb, this is a 10-to-1 ratio. So the peak to dose is 10-to-1. So this will be 100%, this will be 5 to 10%. So it's very, very low. It's a very big sparing effect, 10-to-1. And keep in mind that in this valley, there will be cancer, which is extraordinary. So here's the example of what happens if the beam is too thick. So if I do a 1 millimeter beam, I literally obliterate any tissue, right? With 140 gray, I literally obliterate any type of tissue. But for some reason, if I give it in 25 micron width, 4,000 gray, so a factor of 25 higher, you can see there are cells here surviving. The tissue is still there and there are cells here, a lot of them in fact. And this was noticed in fact in 1961, in a study from 1961, and they even did a study of what size this transition occurs. So as the beam size gets smaller and smaller and smaller, there's suddenly a transition point where the tissue becomes super resistant to radiation. No one really understands why, but this point is in the order of 50 to 100 micron. So it's very, very small. So your beam has to be really narrow for this to happen. Okay, but the tissue becomes super resistant to the radiation. Here's an example of a irradiation, a microbeam irradiation of a piglet cerebellum, and you can see the stripes of the radiation right here. Right? And the pig was totally functional. It was given a very high dose of 625 gray, and the pig was totally functional after 15 months. No tissue damage, the cerebellum was totally fine. And this was for a class of 20 pigs. So they were totally working. So this is a clear indication that mini beams does not destroy the function of the organ. And here's an example of what happens from a DNA perspective. So gamma H2AX, I'm sorry, this should be a gamma, not a P. Gamma H2AX is a marker for double strand break. All right, so double strand DNA break. And here's the microbeam, and the gamma H2AX marker is in pink or red. And you see there is a huge amount of double strand breaks along the beam, but there is none between the valley. So in the valley, there's very little double strand breaks. All the double strand breaks is concentrated in the microbeam. So here again, a lot of double strand breaks, no double strand, very little or no double strand breaks. And here's when you give a homogeneous beam, you have a lot of double strand breaks. So somehow the damage is done only along the beam, and somehow it's affecting the tumor that's in the valley. Here is the possible way of implementing clinical delivery. So there are two main philosophies. One of them is the one on the left, where you have fractionated, especially fractionated beams. So you spare the organs in this region, which eventually will overlap in the tumor. So we'll have a homogeneous tumor, right? So this will be flat, but this will be fractionated into hot peak and valley dose. An alternative is to do the spatially fractionated. So you have a full spatial spread all the way through the patient. This is not so easy to do with protons. It's much easier to do with something like helium ions or even with X-rays, much easier with X-ray. Protons have this slight blowing up, as you can see here, the Bragg peak. So the protons would have to have a large energy to go through all the way without having the multiple scattering effect. So I'm going to mention one of the most important studies in this field by Yolanda Prezado's group, which was published in 2018. And what they did is exactly this. They irradiated a mice which had been implanted with a high-grade glioma in the brain, and they irradiated with a spatially fractionated beam. So you see how the peak and valley, roughly 10-to-1 ratio. And you see it goes through the whole beam, the whole patient. And this is a proton beam study. And they had 4 study groups. They had the tumor bedding mice, rats that did not have radiation. They had the tumor bedding with proton mini beams. And then they had the normal rats as control. And the funny thing is, if you compare the two, the rats with the tumor, which die very quickly, and the rats with the mini beam, with the tumor and irradiated with the proton beams, you'll see there is a significant amount of rats that survive. So high-grade glioma is deadly. It will kill the mouse or the rat very quickly. For some reason, this method of mini beams has managed to spare 22% of the mice. So they have come out completely cured. And the others had also an extended life. So this is amazing. This mini beam or microbeam approach is sparing the person and also curing the cancer. So this is one of the big finds in this study. Recently, my group has developed a mechanism by which this could be possible. We've just published it. It just came out, in fact, 1 week ago. The mechanism is centered on this concept of dose and peak to valley dose, peak to valley ratio. So the idea is very simple. We decided that the dose is not an indicator of what's going on at the biological level. So you see the dose here is high and low, high and low. But what might be an indicator of the effect might be the diffusion of the radicals that are produced when you radiate the medium. You produce all these different radicals and they all diffuse. And the one that happens to be very toxic is this hydrogen peroxide. So we follow the hydrogen peroxide over time. And you see here on the left, if you radiate a small point, it's diffusing and you get a spread of the hydrogen peroxide. And hydrogen peroxide is very stable. So it's a highly oxidizing element that is extremely stable. And so we started with the concept that it's not dose coverage that's important, but H2O2 tumor coverage. So although the dose has the peak to valley ratio, the H2O2 does not. It produces a homogeneous dose distribution. And so under these conditions, we investigated and we found that, for example, hydrogen peroxide establishes a very quickly a steady state level. So after a few nanoseconds, it's already steady state. That means you produce like a tsunami. You drop radiation into the patient, and the tsunami, which is this H2O2 wave, propagates out and fills up the valleys. And so you have a uniform coverage of the tumor based on H2O2. So, sorry. We then looked at all the published data, including Yolanda's work, Dubrovsky's work from recently 2020, and some papers relative to the study in Grenoble from 2008. So there are 4 experimental works out there with a lot of information. And we made a prediction of how long it would take to fill the gap. So you have these dose valleys that you have to fill with H2O2. And our model predicts around 2000 seconds for Yolanda, 14 seconds for Dubrovsky, and a few seconds for the Regnat studies, 3.5 and 0.7. These were the model predictions of how long it takes to fill the gaps, the dose gaps. So basically, I'm filling these gaps with H2O2. And the measurement times were 2100, so very close, 300 seconds for this one, and 1 second and 1 second. So effectively, these 2 were larger than this one approximately, and 1 is larger than this. So these 3, you get tumor control. So if you look carefully at the last 2, which are the most important ones, in 1 second was just enough time to cover, right? Because you needed only 0.7 seconds to cover the valley. But here you needed 3.5 seconds, and 1 second was not enough to cover the valley. So here was a failure, which we predict in the model, and here was a passing. So we had from the same experiment with different gaps. So these are different widths, so 200 micron and 100 micron microbeams. We predicted that the ones that could achieve uniform H2O2 distribution would be treated, and the ones that could not would not be cured. And it ended up that there was a huge difference. And this one, they had 30 mice treated with this approach, and only 2 were cured. And in this one, they had a significantly higher cure rate of almost 50%. So for us, it was really good that we could predict the cure rate. So, like I did before, the major physics challenge for mini and microbeam is presently it's not possible to do commercial proton therapy machines. It's not really possible to do with microbeams because of the fact that you have to have this very thin shaping device, which would add a significant amount of things to the work. And also the pencil beams are not small enough to allow you to do pencil beam scanning in the form of a microbeam structure. And so it's possible that in the future, as the pencil beams become narrower and narrower with the technology, it might be possible to do mini beams. So the microbeams is really impossible at the moment. We do not have micro pencil beams, but we do have pencil beams with small sigmas. They are still too big for this, but maybe in a few years with technology, we might be able to do mini beams with pencil beam scanning. And another thing that is important is we have a model, but it still has to be validated in a new experiment. So we're looking to do an animal study to check this, and we will probably collaborate with other groups on this. Okay, so these are the main challenges I see in the future for both flash and microbeam. And I would like to show you a picture of Heidelberg. This is a picture of the famous Heidelberg Castle and the river, the Neckar River with the bridge. This is a picture of my group where I'm sitting here. And most of the people that did the biology work in my group are Luca and others here. And Maria that did the biology. And I would like to thank you for your attention. Any questions?
Thank you very much, João, for an excellent presentation. We have about 10 minutes for questions and answers. So who would like to start? You can also type it in the chat and then I can read the question.
So from my side, I would also like to suggest that people also listen to the flash talk. It will be later. It will definitely be more clinical, I suspect. Thank you for the question. It's really a good question. So let me just repeat the question to everyone. In microbeams, how does the H2O2 spread work for tumor, but still spare normal tissue? Well, the key difference is that the tumor and the organ at risk have a completely different ROS level. So tumors are very well known to have a very high ROS level. They have much higher ROS. And we believe that this difference in ROS behavior and antioxidant behavior is what's really creating this differential between the tumor and the normal tissue. Exactly how, we're not sure, but we have to keep in mind that the tumor and the organs have a completely different, let's say, ROS level. Okay.
So I would take advantage of asking you a question. When you say microbeam, how wide or how small should the beam be optimally?
So microbeams can vary between a 50 microbeam and up to 200 microbeam. Usually, I think the biggest is in that order, 200-300 microbeam. But you need a very large valley for that, which can be 4 times that usually. So it can be 800 valley, 800 microbeams, 800 micrometers. And the beam itself is usually very small, 50, 100 microbeams. It really depends how you put it, but you need a large valley as you increase the peak width.
And this would be a beam to be delivered using a gantry or a fixed beam?
You could do both. The problem is the pencil beams at the moment are not capable of that. I think the best pencil beam I know is in PSI. It has a sigma of 3 millimeters. Yeah. So it's impossible. And that's a sigma. So it's really difficult. You would need something smaller than that.
Yeah, a few more quadruple lenses, I would imagine.
Yeah, exactly. You would need something new.
This is the reason why I was asking about the gantry or the fixed beam, because for a fixed beam, it should be technically easier to handle.
Absolutely. In Boston, the beam sigma was so big that my former boss didn't even call it a pencil beam, but a brush beam. So it was a different But okay, it's a different philosophy.
Please, more questions from our participants or from the other panelists. Okay, that does not seem to be the case. So thank you again, João, for a very good talk. Since the topic is medical physics and we have these two sort of sorry, another question. I had another question, but it disappeared. No, it's here. In microbeams.
My fault. Sorry, my fault.
Sorry.
So it will be very difficult to deliver for moving tumors like lung targets, correct? Yes, this is correct. it's really important to maintain the peak to valley ratio. If it's moving, this peak to valley ratio gets blurred by the motion and you no longer have this sparing. So no, you don't want to treat moving tumors.
Okay, so we are on schedule. The next speaker is Professor Mohan. He is a professor and medical physicist at the University of Texas, MD Anderson Cancer Center in Houston, Texas. He received his PhD from Duke University in 1969. Prior to that, he received BS and MS degrees in physics from Punjab University. The current research interests of Professor Mohan include adaptive and image-guided interventions in radiotherapy, proton radiotherapy, intensity modulated and X-ray and proton therapy, and radiation dose response assessment. He is a recipient of the Astro Gold Medal for outstanding contributions in the field of radiation oncology. Professor Mohan, thank you very much for being with us. The floor is yours. Or actually,
thank you.
The screen.
Can you see my screen?
Yes.
Okay. I assume you can hear me too?
Hear you too, yes.
Okay. Well, thank you very much for the invitation and kind introduction. I'm going to be talking about clinical or medical challenges and opportunities of proton therapy. And by challenges, I mean the obstacles or the difficulties we have in trying to exploit the full potential of protons. And by opportunities, I mean the ongoing and future research that we need to do in order to achieve that potential. So, if I may, just a very brief overview of proton therapy. In principle, in theory, proton therapy has tremendous potential, but I think it has yet to be tapped adequately. Secondly, protons are very different from, although Dr. Seco indicated that they're very similar, but there are also substantial differences between protons and photons physically, biologically, and also something, the new thing that we are discovering immunologically. And understanding of these differences of critical importance, and in order that we are able to exploit this potential. Thirdly, the type of challenges that, I'll give you some examples of that. The anatomy changes perturb proton dose distributions more so than they do photon dose distribution. In other words, proton dose distributions are more sensitive to these changes than our photon dose distribution. Although the biology, although the relative biological effectiveness, RBE, is assumed to be a constant of 1.1, there is increasing understanding of it, and it is variable, and there are substantial gaps in our understanding of the true biological effectiveness of protons. In other words, this is the old phrase, what you see is what you get, but this is, in this case, what you see on a treatment plan is not necessarily what a patient gets. There are significant differences, and that can have impact on outcomes. Such factors have limited our ability to exploit potential of protons and obscure the real benefits of proton. Although I think that we are seeing some gains, but I think there are also more risks than we may have anticipated early on, and there are a lot more possibilities than we have been able to achieve so far. And as I said, clinical results are mixed. Some are good and some are not so good. And thus there are tremendous opportunities to enhance proton therapy therapeutic ratio through ongoing and future research. So protons or proton therapy derives the potential it has, or the from its physical properties. And as Dr. Seco explained, they have the ability, because they can deliver lower dose at the entrance and virtually no dose beyond their range. They can produce very compact dose distributions, as illustrated in this treatment plan for nasopharynx. On the left is IMRT, on the right is IMPT, and as you can see, there's much lower dose outside the target volume. And then they have different biological effects compared to proton, and those are also derived from their physical properties. And they have different immunomodulatory effects, which I'll address later in the talk. So what are those properties? Well, protons ionize more densely than photons do. They lose energy continuously. And as they lose energy, first of all, the ionization density and the rate at which the energy is lost, in other words, the LET, linear energy transfer, increases, and that increase is also increasing. So the rate at which the energy is being lost increases as a function of depth. So as protons slow down, they increase, the rate of energy loss also increases. And biological effect depends upon the ionization density, so that also increases. So here, what you see on the right-hand side top graph is that the dose is in this monoenergetic Bragg peak. The LET goes like this. The ionization density, or LET, is increasing very rapidly, especially near the end of the proton range. And so the biological effectiveness is not a constant 1.1, but maybe increasing, especially again near the end of their range. And protons stop abruptly when they have lost all their energy, which is a good thing, because that prevents them from delivering dose beyond their range. But at the same time, that abruptness makes them more sensitive to variations in anatomy. So that's what makes proton therapy more vulnerable to changes in anatomy in the beam path. This has been explained that there are two ways of delivering proton therapy. One is passive scattering, and another one is active scanning, or IMPT, intensity modulated proton therapy, delivered using scanning beam. Clearly, this is just an example illustrating that the power of IMPT. What you see here is the three-dimensional conformal radiotherapy with photons, intensity modulated radiotherapy with photons, and passive scattering with protons, and intensity modulated radiotherapy with protons. And as you can clearly see, the dose distributions are the best with IMPT, and in decreasing order, PSPT and then IMRT and 3D conformal therapy. So intensity modulated radiotherapy is much more powerful because of its ability to control the energies and intensities of individual beamlets. And once they are optimized, we can maximize the dose to the tumor and minimize the dose to normal tissues. And that is compared to passively scattered proton therapy. But it has high gradients within the fields, and so it is more sensitive to uncertainties in range and biological effectiveness, positioning, and so on. As I implied, that anticipated potential proton versus photon is yet to be realized. And in the beginning, when I got into the field about 15 years ago, there was a lot of talk about that it's going to be a cure-all, that we're going to see few, if maybe no toxicities with protons. But that does not seem to have happened. And I think perhaps there were some excessive expectations. One, I think, basically what I've said is the greatest sensitivity of high gradient, so proton dose distribution, motion, and anatomy changes, and gaps in our knowledge of relative biological effectiveness of protons, and still immature and evolving treatment delivery and planning systems and techniques. And the other point that I should emphasize is that the knowledge that we have gained from many decades or perhaps 100 years of experience with photons is not necessarily extensible to proton therapy. And there are already examples in the literature that say that we need to come up with newer, better models of prediction of normal tissue complications and tumor control because of such differences. We need to understand the biology of protons better. We need to understand their impact on impact of other uncertainties in proton therapy in order to exploit their potential. Another point is that because of high degree of heterogeneity in patient population, say for example genetic factors or comorbidities or other factors, and treatment techniques, the error bars in the outcomes analysis are large, and they are obscuring the potential advantages of protons. We are not able to see them, or we are not able to see, for example, the effect of variable biological effectiveness. We continue to assume that RBE of 1.1 is okay, but if we were able to isolate these factors so the patient and treatment heterogeneities are reduced, then we may be able to see some of these things and perhaps increase the benefit of and make changes in our techniques and technologies and achieve greater benefits of proton therapy. And of course, the high cost. Now you'll see in the next talk the impact of uncertainties related to motion and those factors in proton anatomy changes on dose distributions. This is just one example. On the left-hand side is a lung treatment plan, proton treatment plan, and what you can see is that the yellow line covers the target adequately. But if the lung tumor is moving, then there are some changes in the dose distribution patterns due to the fact that the tumor is affecting the range of protons. So these are this is just an example of the kind of thing that can affect proton dose distributions and their effectiveness to cure or adequately reduce toxicities. As I mentioned, there are gaps in our knowledge of biological effectiveness. More recent experiments are showing that, in fact, the proton RBE is not fixed. And let me just give you one or two examples here. RBE is assumed to be 1.1 for every situation, and it may have an impact on outcomes. In reality, RBE is a function of proton LET and energy spectra, dose per fraction, tissue and cell type, alpha beta ratio, endpoint. And over the last decade or so, it's becoming more and more accepted that RBE is variable and that we should be taking that into consideration in our treatment planning. Here is one example from our institution. Two different lung cell lines were exposed to protons at different LETs, same dose. I should say these to come up with the same same surviving fractions. And what we see is that the RBE is increasing slowly at first up to the Bragg peak and then very rapidly at the end of the Bragg peak. And anywhere from below 1 to approximately 1.2 at the 1.3 at the Bragg peak, and then all of a sudden all the way up to 3 or 4. Now this will vary from cell line to cell line or tissue type to tissue type. RBE is also a function of dose. This is an old experiment. This has been known for quite some time. And this is from the work of Wooters et al. in 96. What you see is that as the dose decreases, the relative biological effectiveness of protons increases all the way up to 1.8. And more recently, work at MGH showed that for different cell lines, depending upon the different DNA damage repair defects, you can have different RBE, anywhere from 0.9 all the way up to 1.7. So RBE should not be assumed to be a fixed quantity 1.1, and that could have consequences. By the way, this experiment was done all in the middle of SOBP, so they're not at the end of the range. This experiment was, these rises are near the end of the range of protons. So what are the consequences of these uncertainties and gaps in knowledge? And I'll see if I can give you some clinical examples. Biologically effective dose distributions delivered is different from what is intended, and outcomes so far are mixed and perhaps suboptimal compared to what was originally expected. I think there is a lot more potential than what we have seen so far. Some examples, in the most recent examples is what I'm going to give. I'm not going to compare, the work that was done in the 80s or 90s, but in the last decade or decade and a half. Some results are positive and negative, or some are negative, but I think the future looks bright, and it's what we're seeing and what we're learning from what's going on, the research that is going on. I think we see some very encouraging results. First, is an most of the examples I'm going to give are from the work done at MD Anderson. This was the one and only completed randomized trial. This is the one and only completed randomized trial. This is for lung, and it was passively scattered proton therapy versus intensity modulated photon therapy. My colleague Xing Liao led this trial, and it was a trial that was done in collaboration with MGH, actually. And the initial expectations were very high. In fact, we were told by many people that we should not be doing such a trial. We already know protons are better than photons. It'll be unethical to do such a trial. Another point that was often made was, why would a patient, knowing that protons are better, will agree to be randomized between protons and photons? But we were able to do this trial, and what we found is this. We found at the end, there was no statistically significant difference in primary endpoints, which were grade 3 or higher radiation pneumonitis. As you can see, there is no difference. And the local failure rates were also identical. There was no statistically significant difference. So what happened with all the high expectations? We were not able to show the protons are definitely better in either reducing toxicities or improving local control. Well, local control, I can assume that maybe we shouldn't expect. We certainly expected a significant reduction in toxicities. The reason we did not expect any changes in local failure was because the prescription dose was the same in both arms. So what might have happened? Possible reasons are that, again, inter-fraction anatomy variation may have affected dose distribution, by simplistic assumption about RBE of protons, and immature technology. In this trial, we used passive scattering proton therapy versus IMRT. Perhaps if we had access to IMPT in those days. This trial was done in 2009 through 2014, and none of the patients were treated with IMPT during that period, although we have IMPT now. And whereas the photon patients had online imaging, proton patients did not. We had X-rays, but not online cone beam CT, for example. And the dose accuracy, those computation models, accuracy was also suspect in the treatment planning system that was used, and so on. So there may be many factors related to technology that may have kept us from doing the best that can be done. And then it was somewhat of a learning curve. We were still learning how best to use protons, and there may be other factors. So, but the good thing is that this trial generated tremendous amount of data that we have been analyzing, and we continue to analyze to learn from it what could have happened, and then use that information to improve techniques and technologies. Another example, that was a negative example. Another negative example is the retrospective study of brain imaging changes in proton versus photons. This was published in 2015 by my colleague. This was for pediatric intracranial ependymoma patients. And the study involved MR imaging changes in 72 intracranial patients, 36 of them protons and 36 photons. This was a retrospective study of matched cohorts of patients. And the interesting thing that we found was that the imaging changes occurred in 17% of IMRT patients, but occurred in 43% of proton patients. However, more interesting is the fact that that the high-grade image changes, which correspond to hemorrhage and focal necrosis, there were 7 of them in the proton arm and 0 in the IMRT arm. So that was quite worrisome that protons are supposed to reduce toxicities, whereas here we see significant increase in toxicities. And again, this is when we started to first seriously think that perhaps we need to worry about the RBE of proton at the end of their range. And so my, at that time, graduate student, Chris Peeler, he analyzed the data. He did volumetric, VBA analysis, voxel-based analysis. And what he showed indeed was that the probability of image change depends not just on dose, but also on LET. And in other words, the toxicities or the probability of image change is a factor of not just dose, but also of LET. And in other words, it's a function of RBE, and that RBE is variable. But there are some positive examples also. This one is from Mayo Clinic of oropharynx cancer, oropharynx cancer, IMPT versus volumetric modulated arc therapy. This is also a retrospective study by Manzar et al. And they looked at the acute toxicities and patient-reported outcomes for IMPT versus IMRT. And there were 305 patients, 46 IMPT and 250 VMAT. And what they found was that IMPT was associated with lower feeding tube placement. So in other words, even before the patient started treatments, about 18% of the proton patients had feeding tube in them. But by the end of the treatment or afterwards, something like 38% of the patients had feeding tubes. On the other hand, for photons, X-rays, initially there were about 15% of the patients who had feeding tube, but over the course of radiotherapy and afterwards, there were 60% of the patients had a feeding tube placed in them. There were less hospitalization 60 days after radiotherapy. For protons, it was only about 7 or 8%, whereas for photons, there were 1 or more hospitalizations for up to about 30% of the patients. So IMPT was able to reduce the feeding tube placement, and it was also able to reduce hospitalization. Other thing that I'm not showing with diagrams here is that 22% reduction in narcotics used for pain, cough, and dyschezia, and patients in general felt less ill and had better swallowing. So IMPT, in their experience, was able to improve outcomes as far as normal tissues are concerned. I don't remember seeing what the outcomes for tumors were, but I think they were comparable. Another recent study, and this is something, a study was completed, I think, last year. This is my colleague, Stephen Lin. He conducted a randomized phase 2B trial of proton versus IMRT for locally advanced esophagus cancer. And the purpose was to compare total toxicity burden, which is really a composite of 11 different toxicities appropriately weighted, and also compare the progression-free survival for proton therapy versus IMRT. There were 145 patients, 72 IMRT, 73 proton therapy. Prescription dose was 54 gray. There was no difference in survival between the two, and perhaps not to be expected since the dose is the same in both arms. Or perhaps it was not related to toxicities. However, the total toxicity burden was 2.3 times higher for IMRT than proton therapy, and post-operative complications for patients who were operated on after radiotherapy was 7.6 times higher for IMRT than proton therapy. So although, excuse me, proton therapy was able to reduce toxicities, there was no difference in survival of these patients. So clinical outcomes are somewhat of a mixed bag, and there are many, many more examples. I just gave a few to you. But I think that in general, it's not clear what the benefits of proton therapy are in terms of improving outcomes. And I think there are many, many reasons. I gave a few of them, but perhaps there are more. But that also gives us tremendous opportunities to learn from what's going on, to conduct research, to improve technology, and then ultimately to achieve superior outcomes. A substantial research has already been going on for the last 10 to 15 years. I've been involved in some of that. And results are gradually being translated into clinical trials and practice, and outcomes are improving. I think still we still have a long way to go to bring protons to the same level, our understanding of protons to the same level as we have photons. And I think then we'll be able to show clear differences between protons and photons. So a couple of ongoing research examples. One thing very interesting that, or at least interesting to me, that I noticed was based on the work of my colleagues, is the differences in immune system suppression between conventional radiotherapy and proton therapy, and the possible use of proton therapy to mitigate this suppression of the immune system. Just a little bit of background, the immune system suppression or lymphopenia induced by conventional radiotherapy is very common across all tumor types, and it is well known, but it is often ignored as unavoidable side effect. And it's also, oh, by the way, the reason is that the immune cells, our lymphocytes, are very sensitive to radiation. And the fact that the protons have a much smaller dose bath, that means they have smaller volumes that are exposed to low and intermediate doses, which will kill lymphocytes, but perhaps spare them for proton therapy. So high grade, and that means there is a possibility that the protons, the lymphopenia will be reduced. So high-grade lymphopenia is also associated with poor disease outcomes. That is also well known. So we observed, and I'll show you a couple of examples, that proton therapy, due to its compact dose distribution, is less immunosuppressive than conventional radiotherapy. So one example is that of esophageal cancer. Again, a study conducted by my colleague, Stephen Lin and his team. In this study, this was a retrospective analysis. There were 504 proton and photon patients, 187 proton and 317 IMRT. We found that radiation modality, proton versus photon, strongly influenced mean dose, mean body dose. And that's what you'll expect. But that mean body dose is also a strong predictor of grade 4 lymphopenia. Grade 4 means less than 200 cells per less than 200 cells per microliter. And here what you see is a combined proton and photon dose distribution. Combined proton and photon cohort. And if the patients developed grade 4 lymphopenia, the survival was poorer compared to those who did not. And here is a comparison of proton versus photon, that proton patients had a better survival than IMRT patients. Even though this was passive scattering, I wonder if we can do better if we had IMPT, patients treated with IMPT. Another example is that of GBM patients. That can proton therapy reduce the likelihood of high-grade radiation-induced lymphopenia? And this was a study I think just published in the last month or so. We investigated the grade 3 lymphopenia, in this case, not grade 4, which is defined as absolute lymphocyte count nadir of 500 cells per microliter. We found that grade 3 lymphopenia is lower in men versus women, and for proton therapy versus photon X-ray therapy. And grade 3 lymphopenia is significantly associated with baseline ALC, whole brain mean dose, and brain volumes receiving 5 to 40 gray. Here's a couple of figures, which this shows that if the baseline lymphocyte count was the same between protons and photons, after radiotherapy, the lymphocyte loss was more for photons than for proton. Similarly, for the difference between males and females, before the start of treatment, the lymphocytes count was essentially the same, but after treatment, females had a poorer lymphopenia than males did. In other words, females had lost more lymphocytes than males did. So conclusion was that sex, baseline lymphocyte count, and modality were strongest predictors of grade 3 lymphopenia for GBM patients, and that proton therapy reduced brain volumes receiving low and intermediate doses and consequently reduced E3L. And possibly that perhaps females will benefit more from protons than males will. And there are a number of, I can give you more examples, but there's not enough time. I think I just want to, in the end, say that there are ongoing trials there are many trials going on, and many of them are randomized. There's in the United States, there's an oropharynx, IMPT versus IMRT. There's an esophagus trial, again, IMPT versus IMRT, and liver trial, lung trial, a prostate trial, breast trial, and these are the ones that I know of. I'm sure there are many others going on in the United States, but I think not many of them are randomized. My hope is that they will produce results and data to help us better understand proton therapy and then use the knowledge we gain to improve predictive models and treatment planning approaches. And they also will allow optimum selection of treatment modality for each patient based on that patient's specific characteristics. And there is a whole list of other opportunities that I can give you a list of. Addressing uncertainties, filling gaps in our understanding of proton biology. Another one is developing personalized as opposed to population-based predictive models of normal tissue and tumor response to proton and photon treatments. I think that will allow us to then more accurately use model-based selection of treatment modality. So the patient walks in and we can say that this patient has these baseline characteristics, and we can develop treatment plans for both protons and photons and use the models to say, well, this patient is more likely to benefit. And of course, proton flash, and you'll hear more about that later. So back to my original message that proton therapy has considerable potential that is yet to be realized. I think we will, hopefully before too long. Protons are very different from photons physically, biologically, and immunologically. Understanding of these differences is critical in order to exploit the full potential. And there are challenges related to anatomy changes and gaps in our knowledge of proton biology. And what we see on a treatment plan is not what the patient actually gets. And such factors have limited our potential to exploit the potential. And clinical results are good, and some are not so good, but they're improving. So there are tremendous opportunities to enhance proton therapy therapeutic ratio through ongoing and future research. And with that, thank you very much again, and for your attention. Happy to answer any questions.
So Radhe, maybe I ask a question?
Of course.
I wonder, these lung studies and the ones that were negative, would there be a follow-up, let's say randomized trial?
A good question, yes.
Because there were passive scattering studies. What about IMPT follow-up maybe?
Right. Let me answer in two parts. One is that there is a trial, ongoing trial at the national level, the NRG trial, lung trial, that is comparing comparing proton versus photon. Many of the participants do not have IMPT, or did not at that time, and there were concerns about use of IMPT because of its sensitivity to motion.
Yes.
However, in addition to the NRG trial, which is, I think, scheduled to be completed this year or next, it does allow IMPT, but does not force you to have IMPT. Whereas there is another trial that is being conducted at MD Anderson by Dr. Liao, which is IMPT versus IMRT. That has a much smaller number of patients, a phase 2 case. And I think she's already, anecdotally, she's not allowed to, because it's a IRB approved trial, she's not allowed to look at the differences. But anecdotally, there is a feeling that indeed IMPT is doing much better than PSPT.
More questions? Let's take advantage of having Professor Mohan.
So Radhe, maybe I ask a second question. I'm sorry, the pediatric study, I was very surprised. I wonder if, I was really surprised.
You're talking about this one here?
Yes, exactly, the epidemoma study. That there
Yes.
wasn't any major difference between them, effectively, and that there was more toxicity on the proton side.
Mm-hmm.
So was it an LET effect, as you mentioned a few times, in the end of the Bragg peak?
Well, you know, that is the current thinking, that we did find that indeed, as this one, that it does correlate with LET. However, the work done at Harvard did not, you know, and I think there's just not enough patients or not enough in-depth analysis. But it does suggest that there is an RBE effect. By the way, we have found some really serious acute toxicities, optical structure toxicity.
Wow.
in many patients, which was not expected. And I think it may be LET or variable RBE that is causing that. But in the last couple of weeks, I have learned another possibility. You know, as I said, the protons they do not suppress your immune system to the same degree as photons do. So could it be that in addition to LET and RBE effect, the inflammatory response to radiation-induced injury is much higher with proton patients than with photon patients? So in other words, if there is injury, the lymphocytes will rush in to repair it and cause increased inflammation, in fact. And that could lead to necrosis and more damage than, so I don't know. But I think certainly that's very speculative, but I think there is more evidence that we're finding that indeed we really should be worried about higher LET. Even if it happens near the end of the range, I think that end of the range happens to be in a small region, you could see some damage that you would not expect from photon treatment, perhaps. So, you know, the fact that there is no reason not to reduce LET in these critical structures. And by the way, it's not just LET, it's combination of LET and dose. And both, in other words, biological effect needs to be reduced in those regions. And it can be, especially with IMPT.
Yep.
But we have to know that it is a problem.
Yeah, definitely for pediatrics. And what was the age group? Was it young adult or was it even smaller than that? Do you remember maybe?
No, I don't remember the distribution, but I think obviously in the pandemoma case, there were pediatric patients and 18 or under.
18 or under, wow. Yeah. Okay, thank you.
More questions, anyone? Also nothing on the chat. So thank you very much, Professor Mohan, for a very interesting lecture in giving us the challenges from the medical point of view. So right now we are going to have a 15-minute break, and then we will have 2 more presentations, again trying to keep this balance between more physics aspects on one side and more medical aspects on another talk. So we will reconvene at 3:45 Lisbon time, and 9:45 US Austin time. Thank you very much and see you in 15 minutes. Good afternoon again. Welcome back to our session. We will start now the second part of the session with 2 more talks. The first one will be on understanding uncertainties in proton therapy, and it will be given by Tiago Madaleno. Tiago Madaleno is a medical physicist at the Portuguese Oncology Institute of Lisbon, and also at Joaquim Chaves Saúde, which is another a private operator in this business. And he currently works in the areas of brachytherapy and external radiotherapy. He has a master's degree in biophysics and medical engineering from the Faculty for Sciences of the University of Lisbon, and a degree in engineering physics from the School of Engineering of the University of Lisbon. Again, we will have 30 minutes for the talk, and then we will have 10 minutes at the end for your questions. So, Tiago, please.
Hello, everyone. Can everyone hear me? Is it working right?
I hear you.
Okay, that's good. Let me just try to share the presentation. Okay, I think everyone can see the presentation. Yes, everyone. Is it? Are you seeing it? Or?
I don't see it yet.
Wait. Here. Yes, I have to do this.
Okay, it's showing up. Yeah. Perfect. Go ahead.
Okay, great. So thank you everyone for inviting me, and also I'm also happy to be here and hearing these last presentations. So as it was said, I'm a medical physicist working at the moment at the Portuguese Institute of Oncology for the past 4 or 5 years, I guess. I also did the master thesis in Bolshoi Institute in a topic of proton therapy. Anyway, I'm still someone is just starting to discover this interesting field of proton therapy. And I hope to give you some highlight and a general understanding of this topic related with uncertainties in proton therapy. So in advance, I would like to apologize if you all hear some loud noises of planes, because I'm not in the best place in Lisbon to do a presentation. But let's start it. Okay. So in one of the chapters of the book Proton Therapy Physics from Harald Paganetti, where is explained how to deal with uncertainties when using scanning beams. Antonio Lomax wrote this chapter. He's a researcher in Polshek Institute. He shows this image, which is an image from Belgian artist René Magritte. And the title is "Ce n'est pas une pomme," which is "This is not an apple." So the point of the artist to show is that this is a representation of an apple and is not the real apple. So although the image is clearly very beautiful painting, this is not an apple, and let's be clear about that. And Radhe Mohan already spoke a little bit about this important topic. So in many ways, the same can be said about those distributions when they are calculated in the treatment planning systems that we have in our departments. We can really see beautiful those distributions, but they are not the real treatment. They are, although they are quite accurate representations of the estimated dose deposition, it's just that, just representations. And it's in fact a point that this is really well known in every radiotherapy department among all the team. But I believe it's really important to be aware of this. Sometimes we see these beautiful images on the display, but we should really we really should know what that means. So I'll start with just some initial considerations, then some topics about uncertainties related with proton therapy, and in the end, the short conclusions about this. So if we shoot a monoenergetic proton beam, this one here, it's a 200 MeV proton beam. And if you shoot it and measure the dose along the path, we can have this curve, this beautiful curve in red, that is displayed here in this graph. In order to widen the treatment range, we can also produce a spread out Bragg peak. This is done summing different Bragg peaks with different energies and an appropriate weight. So the range will be then defined as the position here, where the dose has decreased 80% of the maximum dose that we can see here in the distal fall-off. So let's head here, as you can see all here in this green profile. Let's head here profile of 15 MeV photon. And as you can see, we had it here, but we have to keep in mind that we have the same integral dose in this region from the 15 centimeters to the 25 centimeters. And we can all see clearly and understand what are the advantages of proton beam when we compare to photon beam. Here in yellow, you can see that this will be the amount of dose that will be not given if you use protons instead of photons. And it's quite clear here the steep fall-off region, and how the protons will give almost no dose in this distal region of the target, and in the proximal region, the dose will be much less than when we compare this with photons. In this graph here, taken from this paper from Paganetti, we can just see the energy relationship of protons and what will be the range in each of these energies. Sorry. So what is then the problem with the range? As I said, the main argument for using protons is that we can have a less integral dose in theory in the patient. And if we analyze this picture from this paper here, it's a representation. And we can see here the spread out bragg-peak plateau clearly covers the target region here in pink red, and it's very beautiful covering region, let's say. And it can spare really well the tissues around it. However, when we analyze a more realistic situation where the uncertainties in the determination of the range are present, we might end up with a profound impact on the actual applied dose. Here in these two figures, the second one and the third one, we can see that we might overshoot the tissue or undershoot the target volume. And of course, if we do that, we can might end up with all the dose that is prescribed in the healthy tissue, or almost no dose in this region distal to the tumor. Of course, due to the characteristics of photons, this is not happening when you use photons. And it's important to understand, firstly, that this is just profiles in water, and of course the treatment is much more complex than that. So it's important then to keep in mind sorry, is one okay. So it is important to keep in mind because evidence suggests that 7 to 10% change in the dose to the target volume may result in a significant change in tumor control probability. And ECFU recommends in radiotherapy that the prescribed dose should be within the 5%. So this is important when we deal in uncertainties in proton therapy. So just to give you an example of what can happen when uncertainties are present. So in clinical practice, this is a prostate case. And in conventional proton treatments of prostate, parallel opposed lateral beams are almost exclusively employed, let's say. Although, of course, we can find some discussions if whether we should use anterior oblique fields or not, or if there's some advantage in that. But this is what is most exclusively performed. So proton therapy. This lateral beam approach is done because it's thought to be least affected by the range uncertainties. This approach, however, when you use it, there is largest radiological path lengths to the target. It might also increase the volume of irradiated normal tissues. Also increase the lateral scattering or give wider dose penumbra than common conformal photon treatments that we normally do in our departments when using photons. So let's talk a little bit about sources of uncertainties in proton therapy. These listed here, some are taken from this paper, range uncertainties in proton therapy from Paganetti, also from the book that I mentioned before. So firstly, clinical. Clinical uncertainties comes from the misdiagnosis of the tumor or some not so well target volume delineation. But anyway, this is more clinical. Let's put this aside and talk about other ones. The spatial uncertainties. They are due to setup and anatomical variations or organ motions. It's important also to keep in mind that in radiotherapy, it's used CT images, and they might have some deformation, and it's also used fusion with MRI or PET, and can have the algorithm or the equipment might introduce some uncertainties. However, with the image modalities that nowadays are used, they are really, there are not so many uncertainties, and this is not a drastic situation. It's really a minor problem. There are also those calculation approximations that are made regarding the CT ounce-full units conversion, the mean excitation energy, called I value, and some tissue density at your heterogeneities. And that the main problem here will be in the analytical algorithms. In the end, some biological considerations that are made, like the ones Radhe spoke in the last lecture about the RP problem. So regarding the spatial and patient setup, so from the point of view of radiotherapy delivery, it's impossible to position the patient every day in the same place, and the patient will not be perfectly still, of course. So the movements of the patient depends on the site that is being treated, the type of immobilization, the patient themselves, and the working practice that is performed in each institute. So this might lead to some range degradations, and when heterogeneities are close to the target, this might be more severe. So it is important to select good beam directions in proton therapy in order to diminish this problem. When we do with organ motion, we should always be taken this into account. Of course, this is transversal to every modality of radiotherapy, whether we are talking about photons or proton therapy. And we can find extensive literature with these techniques, with techniques that allow us to deal with this problem. Here you can just see a representation of organ motion and what can happen when you use protons. The range degradation will be much more severe than with photons. So it should be avoid beam directions that require protons to stop directly in front of some critical structures, for example. So regarding the CT conversion, as you all know, the CT imaging remains the standard image method for radiotherapy treatment planning, because it can provide spatial accurate map of patient anatomy and together with quantitative tissue measurements. These quantities, the CT value, known for ounce-field units, with a range from -1000 to air, 0 for water, and some values, other positive values for tissues or bones, and they're the most tissues that we have in our body. Just to mention that these ounce-field units are derived directly from linear X-ray attenuation coefficients. And they can be correlated with electron density from known materials. And this is the calibration curve you see here in this figure that normally is implemented in order for the algorithms calculate the treatment and the dose distributions. These calibration curves are characteristics of each scanning or CT we have. What happened in proton therapy is that in proton therapy, the CT is also the standard image that is used. However, proton stopping power values must be deduced from the ounce-field units. So treatment planning with protons relies on robust and correct calculation of the stopping power values of the patient tissues. Which are then used to calculate the range of the protons. The two main parameters from this equation, that is the one used to calculate the proton stopping power values, are the density and the mean excitation energy here. So there is, however, no direct calculation of the stopping power ratio values for a voxel based on ounce-field units. So the treatment plan is typically determined based on stopping power maps, which are calculated from the patient planning CT using commonly a scanner-specific stoichiometric calibration curve, like this one here. This calibration method is based on a well-known paper from Wu Schneider from the year of 97. So in order to establish this curve, it involves then computing the stopping power values of several reference human tissues, of which the composition and densities are found in the literature. So there are inevitable uncertainties associated with these curves. The actual conversion is dependent on the chemical composition of the material, and not just in the ounce-field unit value. So, for example, if two materials with the same ounce-field units have the same value, they might have quite different proton stopping powers and vice versa. So the pure uncertainty, excluding the high value of stopping power conversion, is presumably around 0.5%, most for analytical algorithms. So regarding this I value, the mean excitation energy value is a material property which depends on the chemical composition of each tissue, and is then used within the backlog formula to calculate the stopping power values. So in one of the earlier reports, the SRU49, the value is 75 eV for water. Other ones report 80, 81.8, and more recent experimental values gives us 78.4. Here it's a study from Andreo, and he has demonstrated that the variation of these I values from 67 to 80 might lead to a difference of 3 millimeters in the proton beam range. In this case, in the peak. This is for a 122 MeV proton beam. However, there are more recent reports. This one, the 90 from 2006, which recommends a value of 78. However, if a difference in the I value between 75 and 80 might result in a from 0.8 to 1.2% difference in the predicted stopping power when we are talking about therapeutic energy range. Anyway, there are, of course, still uncertainties in tissues based on material composition. So it can be expected a range uncertainty in the tissue of around 1.5%. This is, in fact, probably the biggest uncertainty when it's necessary to calculate proton range. So, how to deal with tissue density heterogeneities? Sometimes we have this kind of structures, especially in the head with skull and hair and soft tissue. And this is, we have these heterogeneities. Also in the lung, we can find these heterogeneities, and they might create range perturbations. So Savakushi, on this paper, he performed a series of systematic Monte Carlo simulations. We can see here represented in these graphs, in order to understand this phenomena of range degradation. So they found that multiple Coulomb scattering is the main cause of range degradation and cannot be predicted using only the stopping power values of the materials that compose the heterogeneities. As you can see by these figures of the mentioned study, the distal fall-off width here, let's see it, increases when the complexity of the heterogeneities is increasing. So most treatments planning algorithms that use analytical models cannot, in fact, account for this effect. They are less sensitive to these complex geometries and might not be able to correctly predict this effect of range degradation. So at the 90% fall off, they found that difference. They found a difference in range about 0.7%. We can then evaluate more clearly in this figure the range degradation. these axial views of those distributions. The first one is calculated using a commercial planning system based on the pencil beam algorithm. And on the left, here on the right, sorry, the Monte Carlo system. In this case, you can you can see here different. Sorry, you can find a scattered disequilibrium here in a small region of the distal dose distribution. In fact, when we are dealing with very small volumes, the effect of heterogeneity can potentially affect most part of the treatment volume. So as Radhe mentioned earlier, you spoke about the RBE. I'll be not so extensive. These biological considerations. So protons, when we are using protons, the same absorber dose as with photons may produce a different clinical outcome. In fact, protons are biologically more effective than photons. So to cause the same biological effects, we need a smaller amount of absorbed dose. RBA is then calculated by this formula, which is then the ratio of the absorbed dose that produce the same biological effect between a reference radiation. In this figure from this paper, it's represented experimental measurements. Here you have the RBE in function of the dose per fraction. The white circles are in vitro measurements, while these here, the black ones, are in vivo measurements. When we analyze these in vivo measurements, especially, which the response reflects the more complex expression of the integrated radiation damage to several tissue systems and various biological processes. The average RBE value represented here in this thick dashed line is 1.1. RBE is, of course, much more complex. It's a complex function, as Radhe mentioned earlier, dose level, proton beam energy, specific tissue that is being treated, the radio sensitivity, and of course, lag. For example, in LET, although the proton LET increases as the protons slow down, the proton therapy treatments are currently planned and delivered, assuming this value, the 1.1 value of RBA. As again, Radhe explained this very well earlier. So there are, in fact, significant gaps in our knowledge of this parameter, which is likely to vary between 1.0 and 1.5 in the relevant dose regions close to the Bragg peak. So the biological uncertainty is around this value, and it should be included this uncertainty in a margin increase for the organs at risk. So this is just to sum up all the uncertainties that I mentioned, more than the numbers itself. It's important to note that there are uncertainties that are independent of those calculations and other ones that are dependent, and then can be different when using different algorithms. So as an example of this, let's say, best case scenario, is if a margin is being added of the order of 2.4% of the prescribed range plus 1.2. adds a substantial dose to the healthy tissue. For instance, if 10 centimeters water equivalent depth in the tissue were required in order to cover the distal part of the target, one will deliver then 10.36 centimeters. It's almost 4 millimeters. So just a quick mention about the how to deal with uncertainties. There are different methods and an extensive literature about that. So it can be similar with radiotherapy using margins, the PTV. Although might not be as linear as photon therapy, because the range uncertainty margins depend on the depth of the tumor, and the margins might be different depending on the beam direction. They are in fact functions of the beam direction, and then the proximal and distal margin of each beam are expected to be larger than the lateral ones. Just check if so I'll quickly finish. Other methods are the robust optimization, which has been established in proton therapy, and it consists in testing the effectiveness of the plan in protecting the clinical target volume from the range uncertainties. To do this, normally the nominal plan is recalculated, let's say, for different scenarios that incorporate uncertainties. For example, moving the CT anatomy with respect to the beam in various directions, or add relative stopping power ratios, different ones, adjusting them, and we can understand which plan is more robust than others. So there are also in vivo proton range verification, for example, using prompt gamma imaging, motion monitoring, like the techniques that are used in conventional radiotherapy, or some other like rescanning or tracking. It can be account for biological effects in IMPT optimization, or for example, it's something that is being studied using dual energy CT in order to estimate the stopping power ratio values. So to conclude this presentation, I'll just like to point to this interesting article from Untitled where he points some myths. The first one, range uncertainty substantially mitigates the advantage of protons. So like I mentioned, even if we accept an unplanned overshoot when we increase the distal margin, only in this distal edge of the spread out bragg peak region with some millimeters, and in the first few millimeters when entering the patient's skin, photons have an advantage over protons. At the vast majority of depths, distally or even proximally, the advantage of protons remains. So, and also another myth is the bragg peak is the sharpest gradient. As I mentioned before, this is what we see when we see these beautiful graphics. Let's say when you shoot a proton beam to water. In fact, there are, because of range degradation, when we deal with the patient, it's not so sharp as we might not think. And proton, another one, photon therapy is robust, proton therapy unrobust, and I will just and it's not always the case because it depends. For example, if you use VMAT and we have ugly gradients, they might be less robust than proton therapy, some proton therapy treatments. So thank you very much for your attendance, and sorry for go over the time.
Thank you very much, Tiago. We have time for a couple of questions from the audience.
So where can I see it?
You can also type it in chat directly to Tiago, who will read it, or to myself, who will read it.
May I ask a question? This is Radhe Mohan.
Go ahead, Professor Mohan.
Of course, of course.
So you indicated, you seem to imply that the range uncertainty is 2.4% is the most important uncertainty. Did I understand you correctly? That 2.4%.
What I would like to mean with that is, like, let's say, just pointing, let's say, from the ones who were shown the best case scenario, let's say.
Yeah, and that you need to add 1 millimeter margin or so.
Yes, yes.
Extra. How about, how does that compare with the uncertainty related to, say, motion or changes in anatomy over the course of radiotherapy, and not necessarily setup uncertainties, but let's say the tumor shrinks or with radiotherapy, or that the lung tumor is moving. Are they more important or less important than the 2.4%? Even with best imaging, yeah.
Yes, yes. So I think from my point of view, let's say that what I know, and I think when you deal with lung in these cases, it will be probably necessary to add margin, the biggest margin.
Yeah, in particular, because if the dose goes beyond, you have to have a margin at the end of the tumor. If that margin is in low density tissue, then the protons will travel very far into lung.
Yes, yes. I think there's also a study from MDA, and I think it's from your team, I guess.
Yes.
This one where you use like a plastic human lung, and then the results show the widening of the dose follow, right? And you show that it's caused mostly of the CT resolution. So this will, of course, introduce largest margins, I guess.
Well, or there may be other ways to take into account, because if you want to, other, yeah. So I think there is a lot to learn. I think in my opinion, then we cannot ignore these changes for protons in particular. Yeah.
I'd like to add, hi. I would like to add that I don't do proton therapy in my department, but from what I know, because of the changes throughout the treatment, for example, due to edema and the reduction of the tumor, you need to overcome these changes that have an impact on the treatment delivery. You need to do more treatment replanning than we do with photon therapy.
You're absolutely right. I think not just edema, but also tumor may shrink over the course of radiotherapy, or that a patient may lose weight. And so
of course,
all those kinds of things have to be taken into account. And I think it is not done regularly. It is done sometimes, but not enough. So if we want to take full advantage of protons, we have to do more of those kinds of things for protons than we have been doing for photons.
Yeah.
Yeah.
Are there more questions? I actually gave wrong information before. I said you could use the chat, but no, you have to use the Q&A. If for some reason you are not able to use your microphone to put the question.
There is a question on the Q&A. Yeah.
What is the real quantitative impact regarding proton range from your study about HU to RSP conversion?
So as I mentioned, the impact of the CT has been probably the biggest one. This conversion, because of the mean excitation energy values that are being, let's say, updated, and also because of the densities and the chemical composition of the tissues. So the real quantitative impact, it also depends on the algorithm. If it's all, I believe it really depends on the algorithm as well. If it accounts for Monte Carlo or if it's analytical. So in this paper, if I'm not wrong, it can be, so it's the 0.5 because of the conversion itself and the I values, which is 1.5. So the real value, I'm not really sure which is, but the main impact is conversion.
Okay, thank you very much, Tiago. There's a lot to learn and about proton therapy, and I'm very happy for that, because we in Portugal, we are eager to learn and also to contribute to the general knowledge on proton therapy and ultimately offer a better healthcare to our cancer patients. So I don't know, I miss a lot a live session where we can applaud. I don't know if it is going to work or not, but I would ask you to applaud with using your cameras and so on, to give a final applause to all our speakers to show our appreciation. Thank you very much and have a nice day.
Thank you.
Bye.
Bye.
Thank you very much.